Hello, everybody. Can you guys hear me? No, Dave, I know that you can hear.
Yes, we can hear online.
Okay, good. Thank you. So today I will be talking about our data analysis platform and how it can help you analyze your various NGS-based data. And before I continue, I wanted to highlight that I have financial conflict of interest in that I am a co-founder of Datirium in addition to being a faculty here. And Datirium has licensed some of technologies from Cincinnati Children's and is commercializing this platform. Before I begin, I would like to give some credits. And the development of the platform and the pipelines that I will be talking about was done both at Datirium and at Cincinnati Children's. Andrei Kartashov was the key developer of the platform, and he moved from Cincinnati Children's to Datirium. Michael developed a lot of pipelines. Valery is developing more pipelines. And we learned a lot from our colleagues in Nathan Solomonis' lab. In particular, we worked with Kairavi on TCR, and from Emily Miraldi lab and Alex Katko, where we have collaboration on multi-ohm data. On the Datirium side, there are software developers, Sean, Ido, Daniel, Nick, and Vicky, and bioinformatician Robert Blair, who are working on this. And the funding to develop the platform came from small business grants. We got phase 1 and phase 2 STTRs from Human Genome Institute, and we got technology transfer funding from the state of Ohio. So let's start with why do we need the platform? What is the problem that we're trying to solve? So most of the biologists are at least reasonable in doing experiments. So we can make libraries, we can send them to sequencing, to Dave Core, and then what do we do with the data? And usually the data go to bioinformaticians who start analyzing them, but there are not enough bioinformaticians. They are working on multiple projects, and it takes a lot of time for the results to come back to biologists and even to figure out whether the experiment worked or not. Then you need to change some parameters. You want, I don't know, 2-fold change instead of 1.5, and it takes another round, which takes another month. More importantly, bioinformaticians work on multiple projects, and usually they are not going to be experts in your particular biological system or disease. So they wouldn't be able to make as good conclusions as the specialist would in interpreting the data. And this is why we want to automate at least the early part of the data analysis process with our data analysis platform, so that biologists can do at least the initial analysis themselves to get quality controls, to figure out whether the experiment worked, to get some images for publications. And then if there is something that's difficult to automate, it's possible to interact with bioinformaticians who will be using pre-processed data to continue the analysis, to answer more specific questions, or to do some special techniques like gene regulatory network modeling, like what Emily does. So the advantages for biologists is faster turnaround time. It's basically as soon as you enter the data, and as the cluster crunches the data, you will get the results. You don't need to spend time learning how to code. You still need to understand what you are doing and basic statistics behind it. The pipelines that we're using are all open source. We mostly don't have anything proprietary. We just make them easy to run. This is basically what everybody else is using. We provide quality controls that help you troubleshoot your experiments. You can easily explore data, change analysis parameters, and get some publication quality images. For bioinformaticians, the advantage here is using pipelines that are transparent, reproducible, and portable. So almost all our pipelines, as I mentioned, are open source, so you know exactly what our platform is doing if you are using our pipelines. If you have a pipeline that you want others to use, you can easily add it to our platform, and we will work with you to make it happen, so that you don't have to run this pipeline for everybody, and people can use your pipeline without you holding their hands. The data are stored centrally together with metadata, and this allows easy reuse of the data, sharing of the data, and continuing to use the data. And by the way, easy upload to the geo for your publications. And you don't need to run around with hard drives because the data are centrally deposited. Now for PIs, the advantage is that they get immediate access to data once they're processed. You can browse it. You can audit the quality of the data and how the data were analyzed. There is continuity. So for example, if somebody leaves a lab, not only their data, but the methods that were used to analyze this data will stay in the lab, and you wouldn't need to reinvent the wheel. And it makes easy for lab members to share the data. I have a couple of screenshots, but I will be going into the platform itself to show you how it works. So to add a sample, you need to select what is the type of sample, in this case, ATAC or ChIP, provide some kind of experimental description, select the genome, and attach a FASTQ file, which can come, you can drag and drop, you can get it from FTP, you can put SRR number to get it from NCBI, and we hope to be able to browse the core facility files eventually. Once the sample is processed, you will get a list of your samples with some basic controls, in this case, mapping stats in the list. If you go into the sample, you can get basic stats, some QC data, you can look at the data on the browser, and you can compare your samples using either visual approach, like this 3 ChIP-Seqs on genome browser, or statistical approach to, for example, look at differential binding, differential expression, and so on. Now, Krish asked me to talk a little bit what we have under the hood, and our pipelines are all based in something called common workflow language. So here I will step back a little bit to give you some background on where these workflow languages are coming from. So as people started publishing more NGS and big data, other people wanted to reanalyze, and so they started looking at the method section of the paper and reading how the analysis was done and couldn't reproduce the data. Then people started putting their pipelines on GitHub, and sometimes it was difficult to run. Sometimes the pipelines were dependent, for example, on the version of operating system and so on, and were not running or were not reproducing the data. We had one case when I think it was SAM tools changed their syntaxes, and when we updated operating system, which started using the next version of SAM tools, our pipeline stopped working. And this became annoying enough so that people decided, okay, we need a way to solve it. And part of the way is, of course, to use Docker images, but also you needed to provide more detailed descriptions of your tools and pipelines than what can be done on GitHub. And so people came up with several workflow languages. One of them is called CWL, or Common Workflow Language. It allows to describe two things, which are basically tools and workflows. So for tool, you describe what are the inputs, what is the command that you are running, the environment that you are running this command in, which is a Docker container, and the outputs that the tool will produce. And the description includes not just the name, but what is the type, what is the meaning of this, and so on. Because we have this very detailed description of the tools, it becomes easy to connect them into workflows. So you connect output of one tool to input of the next tool. And this made it much easier to assemble the pipelines. And in fact, there are graphical editors where you can drag and drop tools. There are alternative workflow languages. We are not using them currently, but some of you might. And it is called Workflow Definition Language, which is used mostly by Broad Institute. And there is another language called Nextflow, which is a private company. So it is kind of semi-proprietary language, but also popular, and they have a number of good pipelines. Finally, SnakeMake, which is another pipeline tool, can export the pipelines into CWL, so we can get them into, we can get SnakeMake pipelines into SciDAP. So we took CWL and we also added what are called CWL extensions. So what extensions are is it allows us to interpret inputs and outputs in order to create interface for the pipelines. So since we have the input information and what is the type of each input, we can create the input forms that you saw. And since we know what is the type of output, we can use a specific viewer for each file that is produced. For example, if the pipeline is producing big file, we would know that it needs to be displayed by on IGV genome browser. Or if it is producing volcano plot, we can use the graph viewer to display the results. One more thing before I go to the pipeline, to the platform itself, is how architecture of SciDAP works. So in order to access SciDAP, you use your browser and you can go to SciDAP.com website. After you log in, you will be connected to what we call satellite server. This satellite server, in the case of CCHMC, is actually on CCHMC cluster. And this way, Datirium is not getting your data. So you go to SciDAP, you tell SciDAP, okay, we need to process this Barski file with this pipeline. So this command is being sent to the satellite server. Satellite server would get raw data from core facility or elsewhere, process the data, and then data will be displayed in the interface here, which will allow the user to view the data without data being exposed outside the organization. Another advantage is we are controlling SciDAP.com, which provides the interface, and this allows us to update the interface and pipelines as needed, so you will always have the latest version available. If you want to see how to use SciDAP, we have a YouTube channel. You can search for SciDAP or Datirium. And we also have a blog on Medium where we describe the use of the pipelines. Overall, we have some 90 pipelines, and they can be used to process data generated by single-cell methods like 10x, RNA, ATAC, and multi-omic. We're adding Rhapsody and TCR. Bulk transcriptomics, paired, single, stranded, various kinds of unstranded. Quant-seq data, microRNA sequencing. For epigenomics, we have bulk ATAC, ChIP-Seq, CUT&RUN, CUT&TAC, DNA methylation, germline variant calling pipeline, and we're also going into microbiome, adding pipelines for both 16S and shotgun sequencing. Next, I am going to show you how the platform works for the analysis of epigenomics data. But if you have questions at this point, it's a good time to ask them.
Well, I have a question. Do these there's different workflow languages? Have there real what I mean? What do you have any idea what sort of the differences are? I mean, they're all of them all seem. I don't know if they're all Docker based. I know Nextflow is and CWL is. I assume.
Yeah. So CWL and WDL, I think they kind of came out from the same discussion, but being broad wanted to be different. And CWL team wanted to be more formal and to make it more like a standard rather than just language. And so they have a very detailed standard description, and I think this helps the use. And it is kind of, as a standard, it is maintained by a group of developers, which is combining both industry and academic people. WDL is broad only, and Nextflow is some weird commercial company somewhere in Europe.
I mean, it's Apache.
Nextflow, yeah.
I mean, they're all open source license. I mean, at least.
Yeah. So
CWL is. I mean,
CWL and WDL are Nextflow, the pipelines, but their main tool is not exactly.
No. Do you have any? Is there strong reasons that you went with CWL, or I assume they're all fairly similar in what they support?
So I think they're similar, but CWL has more detailed and standardized description, and we need more standardized description for creation of our.
If you're going to have a standardized workflow language, it probably should be standardized. Yeah. Okay. Thanks.
Okay, so this is actual interface. So we are on our demo server. In SciDAP, the data are organized into projects, and projects includes both data and the pipelines that are used to process it. And this is to avoid comparison between apples and oranges. Data processed with one pipeline versus another. If you go to edit project, you can attach pipelines to the project from either Datirium's pipeline library, or you can connect your own GitHub if you want your own pipelines. So to add the sample, you use this wonderful add sample button. You can select the type of experiment, in this case, ChIP-Seq, drop down, you provide the description, you select the genome, and you can use a file manager to attach your raw data. So here you can just drag and drop the sample into drag and drop your FASTQ files. You can use FTP connection, or if you have a direct URL, you can put the URL, or you can put SRR number if the data you want to process are in GEO. Once you do this, you can click Save Sample. And this will start the process of mapping and processing and so on. So let's look at the results. So here, for example, we have RNA-Seq file. So RNA-Seq sample. So here you can see once it is processed, you get statistics for mapping. You get adapter trimming, alignment, and statistics, and so on. You can look at the results on the IGV browser. And since these are strand-specific data, you get two tracks. The reads actually mapping to the strand that's opposite gene expression. So actin B goes from right to left, and we get reads on the top strand from the UTP. This is IGV browser, so you can do all the normal stuff, zoom out, go to another gene, and so on. We have a number of QC plots, in this case, base frequency. So I personally like to see that AT and GC in RNA-Seq is more or less similar content. You get quality control for reported by the sequencer for each base, and you get gene body average tag density profiles. So this can tell you whether your RNA has been degraded. Of course, you get a gene expression table, and we actually calculated 3 ways. First, we start by calculating gene expression for gene isoforms, and then we can sum them up by either gene or by common TSS. And I guess this depends on what you are studying. So, for example, if you are studying promoter activity, then common TSS makes a lot of sense. If you are looking more at gene function, then you want to look by gene. All the files produced by pipeline are available here, and you can look at the workflow in this workflow tab. I will briefly show you also ATAC or ChIP-Seq sample. The results kind of look similar. Okay, let's look at, so this is JUNB ChIP. Again, you get basic stats, streaming, alignment, peak calling, and so on. For QC, you get, again, base frequency plot, and you can see unlike RNA, you have the genome is 80 rich. QC for the sequencing itself, average tag density, JUNB is enriched around the transcription start site. This one I like very much because it shows you distinct read estimates based on how much you sequence. Basically, it will tell you whether you will be getting more distinct reads if you do more sequencing, or you will get more peaks. You get a genome browser. So let's go to gene. So you can see the peaks, and this is our peak calling using Max tool. You get the list of the peaks that we called in the table. It also shows what is the nearest gene, as well as where it is located relative to this gene. So, for example, upstream, promoter, and so on. Okay, let's see. You did your initial data processing, and now you want to compare the results. So you can go to this sample comparison tab. Okay, let's find some samples. So yeah, This compares, here, for example, you can view both ATAC and RNA-Seq samples on the same browser. You can also compare the technical features. For example, this will be on the fly QC table. So here it will look at what are the reads, what percentage of mapped, unmapped, and so on. And you can also use this on the fly QC plots to compare, for example, enrichment between these two ATAC files, or if we had more than one RNA, you would be able to see this degradation. Okay, so this visual analysis is cool, but doesn't cut it for most of the publications. So you need to do, of course, statistical analysis. And for this, we provide tools such as DSEQ, DeepBind, and MANORM for differential gene expression and differential binding. So let's look at DSEQ. So to run DSEQ, you basically need, we have both pairwise and multi-factor version. So pairwise compares two conditions, and the multi-factor allows you to compare multiple conditions and set up different kinds of comparisons, again, using DSEQ. So I'm going to show you pairwise. So to start it, you provide the name of the sample. Use check mark to select the samples that you want to compare for condition 1 and condition 2. You can select whether you want to do it by genes or by common TSS or by isoforms. There are some advanced parameters where you can, for example, provide the name for conditions, select how to make clustering and heat map. Once you run it, you get, of course, differential expression table, which will provide for each gene initial RPKMs, false changes, p-values, and adjusted p-values that you can use to filter your gene lists. In addition to this, we provide a number of interactive plots. One is PCA plot, and this can be used to look at the samples. So in this case, we are looking at naive and activated T cells. So this is control versus treatment, which is the main difference between the samples, dimension 1, which is 84% of the variance, and the donor is the second dimension, which is 10% of the variance. So in this case, we can see that our replicates are consistent. We can look at the volcano plot, and you can change a lot of things here. For example, you can highlight, let's say 10 genes, 10 most differential genes by p-value. You can change the colors. And so on. So, and you of course can export the data as either SVG or PNG for your publication. MA plot works similarly, and you also get a heat map which includes your differentially expressed genes that has been also clustered. Once you do this analysis, you can, for example, make gene lists. You can run a gene set enrichment analysis for which DeepBind experiment would be serving as input, and to identify the pathways. Similarly, we have a DeepBind experiment where you can compare differential binding. In this case, it is actually differential accessibility between resting and activated T cells. So it produces a number of QC graphs. So this shows you correlation between conditions. There are PCA plots, MA and volcano plots. You can look at the results on the genome browser. So this will show you which peaks are called in each of the replicates and which peaks are differential. So for example, IL-2, you can see that the peaks at the promoter and enhancers are differential, and you can confirm it visually here. And these are originally called peaks. And of course, you get a table which would list differential peaks, p-values, fold changes, and what are the nearest genes. Based on this, you can create lists of peaks, which can be inputs, for example, for motif analysis, or to build various heat maps and plots. Okay. Do you guys have any questions about the bulk data? If not, then I will move to single cell data analysis. Okay, so these are pipelines developed by Michael, and he also developed nice videos, which I will be showing in the presentation. Wouldn't need to go into the platform itself for the sake of time. And this kind of manual is currently on bioRxiv, and it will be published in the Methods in Molecular Biology book series in the few months. So let's start. So the paper describes analysis of multiome, but you can also use this guide for analysis of single-cell RNA-Seq and ATAC-Seq data. You just need to skip some of the steps. Why use multiome? So initially, as you know, 10x developed single-cell RNA-Seq and single-cell ATAC-Seq, and this allowed people to look at what genes are expressed in each population and hypothesize how these are regulated. And hypothesize refers to the fact that the linkage between the groups of cells, RNA data, and ATAC data was hypothesized. So likely these are the same cells, but we are not absolutely sure. With multiome, we can actually get ATAC and RNA data from the cells, same cell, and the linkage between epigenome and gene expression is a real true linkage. So the way they're doing is, the experiment is you take your cells, prepare the nuclei, take them to our wonderful core facility. The nuclei undergo transposition, ATAC reaction, and then they are mixed in the 10x chromium controller with the beads and oil, creating these so-called gems. Then gems are RT'd and barcoded, and you create two libraries, one for ATAC and one for RNA. So these libraries, GX refers to gene expression, are sequenced, and we need, for gene expression, we need two FASTQ files, read1, which would contain cell barcode and UMI, and read2, which will contain the actual fragment of the RNA, and this lists how long these guys should be. For ATAC, we need actually read 1, read 2, and one of the indices. And this would be the index for a given cell. So the examples are based on this paper that was published some time ago. And the data are, of course, in Geo. And so we were able to download the FASTQ files for RNA and ATAC. And then we used it as an example here. So first step, of course, is to create a project, and where we need to add our pipelines. And for pipelines, we have, Misha came up with this board game type picture, where you start with your data, whether you have RNA and ATAC, or just RNA, or just ATAC, and you go through all these steps to win. So we have a table with the pipelines in the preprint, which you can use to select the pipelines you need. So let's start with adding a sample. So it looks almost exactly the same as I showed you for the bulk samples. You provide the name, select the genome, and attach the FASTQ files. Files can be attached by all the methods that I discussed before. This is how the output will look like. So you get the quality control reports, FASTQ, FASTQC, as well as 10x, and you can also view the data on a built-in UCSC cell browser. So this is Cell Ranger summary. It has, for those of you who looked at the data, you can see it's very familiar. So it gives you preliminary clustering. It gives you some QC for ATAC. So this is fragments per cell, peaks, enrichment of ATAC around the TSS, size distribution. And for gene expression, it shows how many reads you have, how close your library is to saturation. On the cell browser, you can look at ATAC or gene expression data. So this is the clustering that was done by Cell Ranger pipeline. You can highlight the clusters, you can highlight the genes you are interested in, and so on. So next is the aggregation step. And this is, so if you run more than one library, you can aggregate them together. So you provide the name, select which libraries you want to aggregate, and you can select whether you want to normalize the results. Once you save the sample, you can look at the Cell Ranger aggregation report, and you can look at the data on the genome browser as well. A note about normalization. So Cell Ranger aggregate offers either depth normalization or no normalization. And depth normalization will will adjust sequencing by removing reads from the better sequenced experiment. So you can use it if you want to look at the immediate results on the loop browser, for example. But it obviously results in the data loss. If you are planning to use Seurat for your normalization and integration of the data, then you should skip normalization. So this is how the steps of analysis work. So you start with the aggregated cells, or if you have single experiment, just arc, cell-arranger arc count output. You do filtering to remove wet cells or empty cells and doublets. Then you do dimensionality reduction and integration separately for RNA and ATAC. And then this is followed by clustering, and you can combine modalities with the weighted nearest neighbor clustering. So the first step is filtering, and you provide some kind of the name, and you can select your the experiment, in this case all samples aggregated, for your filtering step. You can provide also some metadata for each of your libraries. So this would give the sample names. And there are, in the advanced, you can select parameters for filtering, for example, how to remove doublets. This is a CW doublet finder. You can select what are the cutoffs for removing cells. And here in the information, you can see the description of these parameters. You can remove cells based on mitochondrial percentage, number of ATAC reads, and number of peaks, and so on. So once you save the sample and it is processed, you can look at the results, and this will show you what cells have been filtered out. So these are PCF plots showing the relationship between different measures, how redundant they were. These are distribution of various measures, and this shows you where the cutoffs are. And this will help you adjust your cutoff parameters. And these are the doublets. We have this for all cells, and we have this for the cell split by the group of samples based on our metadata. So, and you can see before filtering and after filtering. So next is dimensionality reduction analysis. So we have some 20-something thousand genes and probably more of ATAC peaks. And looking at all these tens of thousands of dimensions is kind of too complicated. So what you want to do is you want to select the genes or the peaks that will be the most informative, and you can select how many you want. So if you select few, you would see only the most significant differences. So for example, cell types. But if you select more, maybe you will see finer difference, but then you can start messing up your cluster distributions. So this step also integrates the data, and we can use either Seurat or SciDAP integration or Harmony integration. So Seurat and SciDAP, what they're doing is they look in each sample and they try to find integration anchors, which would be the cells that are likely to be the same, and they make sure that these cells would be in the same place on the map. Harmony puts them together in a different way, and it can try to remove influence of the parameters that you don't like to bring all the cells together. So this is the video of how to run this analysis. Again, start by the name, select the filtered cells as input. There are several methods for normalization and integration, as you can see here. You can, if you are using Harmony, you can also correct for batch effects. And you can provide the batches information in the future steps. Here you can also either remove or partially remove cell cycle effects. And this is, I think, the metadata. And you can see that this provides the metadata information. In the advanced, you can select parameters of how many variable genes or peaks you want to get, whether you want to show the data to create a UCSC browser view or not, and so on. So this is why integration is important. So here, if you just, here we have data from for donors. And if you just merge the data, you would see that the donors are not, that each cluster, that the cells are clustered based on the donor rather than on the cell type. If you integrate, you can see that also that each of the resulting clusters is represented by all the donors. And you can see the easier find the cell types. So these are results. This is the elbow plot, which allows you to select the number of dimensions in your PCA. You can see the UMAP. So this is how well the data were integrated. You can color by group, by data set, and so on. And you get UCSC cell browser view, which will contain the cells, and you can color by patient. So this will check the quality of your data integration. You can color, yeah, this is by patient. You can color by QC parameters. So this is mitochondrial, number of genes, and so on. And this is cell cycle phase. So you can find dividing cells. Next step is clustering. We want to separate the cells that we previously saw in the UMAP space into cell types. And this is done with the clustering pipelines. So we have our input as a dimension reduced experiment. We again look at the number of PCA dimension, and we can select clustering resolution. We can also add a number of genes of interest that we think will be important. And this would allow us to get additional plots and information for them. If your data are 10x, our pipeline can produce also loop browser so that you can look at the results not only in our browser, but download it and use CLoop. So these are the outputs for the genes of interest that we selected. We get we can get clustering, and for each cluster we can get expression for the genes we selected. Let's look at the video. So this is QC information for each cluster. You can see whether the clusters are driven by technical or biological. Above this, there were silhouette plots showed whether the assignment is good. You can see composition of clusters, and this highlights the gene expression for your genes of interest in the UMAP plots.
Can you maybe show how can you tell that the change is driven by a technical or a biological effect?
So if you so this shows you the distribution of the technical parameters for each of the cluster. You can see that, for example, this green cluster has more reads than some of the other cells. And so this probably should make you question whether this cluster is separate because the cells have more reads, or because it's a real biological cell type that just has more RNA in it. And so you will be able to look at it. More damning would be probably mitochondrial percentage. And so you will be able to see how this is distributed on these violin plots, as well as in UMaps on the browser. And again, you will be able to ask yourself the same question. Is it reasonable to think that this cell has a lot of mitochondrial RNA, or maybe it is a dead cell and all the RNA has flown away and we only have mitochondria left? Okay, I think it was somewhere here. So we have various plots for our gene of interest. So these are violin plots for each gene. We have dot plots. We have a heat map for markers that were identified automatically on the table for the markers for each of the clusters. And all the files again are here. So I'm going to skip the steps for ATAC dimensionality reduction, as well as clustering and WNN clustering. And WNN clustering allows you to combine information from both of the modalities, and can better, for example, for T cells, can separate cell types much better. You can also do manual cell type assignment. We also have pipelines for trajectory analysis for immune profiling. So this would identify the cells that have the same TCR or BCR sequence, and you can look how are they distributed between T or B cell types. And probably that's it. We have a few minutes to answer the questions. And we have free trial. So if you are interested in trying SciDAP, drop me an email, or you can email to Datirium at datirium.com, and we will set up a demo for you, and we will have somebody show you how to use the platform. And as I mentioned, the demo is free. If you are interested in subscribing, we will install it on the cluster, and you will be able to use this at CCHFC. One more thing that I didn't mention is probably important, is the outputs of this pipeline. In addition to the visual stuff that you saw, all pipelines produce RDS files with the Syrah object that you can use for data analysis. We'll also produce Loom files, which can be viewed in the Scope browser. And for the next data, we produce a CLOOP file for the next Loop browser. Okay, any questions?
Yes, I had a question. I mean, it sounds like you're using a mix of R. I mean, I guess on the back end, is it R, Python, both, everything?
Depends on pipeline.
Okay.
Everything that is related to single cell was written in R.
Okay.
But for RNA, bulk RNA, bulk ATAC-Seq, like the first part of the slides, it could be any other application.
Okay.
R, bulk type.
Yeah. Where do you guys stand on like moving to like some of the more Python-based workflows that are maturing more, but probably behind this?
So the workflow itself is WL. Each of the steps can be R or Python or Bash, whatever.
Yeah.
And I can give one more detail. So the reason why we chose R, because tutorial for CUT was way more better explained than anything similar on Python.
Sure. Yeah, absolutely.
That's why it was like obvious choice.
Yeah.
At that time. Maybe now Python is better.
Yeah, I mean, we had a talk several months ago about some of the tools in Python. It looked like they're coming along quite nicely. I'm sure they're still lagging behind R, but it looked very promising for sort of a more performant, more integrative.
A workflow that runs Python package. That package is written by a postdoc from Nathan Solomoni's lab. It's called the C-Triangulate, but we still run it from R just to make everything like consistent.
Yeah, but thanks to CWL, we are kind of pipeline, we are language agnostic.
And you should be, but I just kind of wondered on the back end. You showed exports to the UCSC cell browser, which I didn't even know was a thing. Now I do.
So it's not export, it is built in. So
Sure, yeah.
It is, single button. It's very convenient to use.
Is it better than cell by gene, do you think?
Cell by gene is difficult to integrate in the platform.
Okay.
And this is kind of very lightweight. You could see how fast it opens. You don't need to mess with it a lot.
Do you have ways to export to think or like have connections so you could load your data into the UCSC genome browser?
Yeah, so we are making big wig file, but for the genome browser, we have built-in IGV.
Okay. You can have a UCSC browser built in too? Oh, that's probably harder to do.
It's harder to do. It's not licensing, it's open source. And in our previous platform, we had UCSC, but it was a lot of work to maintain.
Okay. Fair enough. Cool.
Nice. Thank you.
Thank you.
Thank you.
Thank you.
Thanks.